3 o'clock in the morning. A child arrives in the emergency room. His mother says, he hasn't been himself lately. During the day, he can still barely make it through school, but at night, he says someone is talking from the corner of the room. He becomes frightened. He hides in his bedroom. He refuses to go outside. Sometimes his eyes suddenly roll upward. His body stiffens. After a fever, everything gets worse. Just a few months earlier, he had only seemed a little slower at school, a little clumsier, a little more tired. If you were his parent, your first thought might be, is this a psychiatric problem? Is it epilepsy? Is it encephalitis? Is he under too much stress? All of those possibilities matter, and doctors have to check them. They usually begin by ruling out the common causes: seizures, infection, encephalitis, medications, toxins, sleep problems, trauma, and severe stress, a first psychotic episode, or a mood disorder. But some children cannot be understood through a psychiatric diagnosis alone, especially when the story includes red flags, developmental regression, abnormal movements, eyes rolling upward, very low muscle tone, clear worsening after infection, clear worsening after fasting, or MRI changes in the white matter or the basal ganglia. At that point, a doctor has to ask one more question. What if this child's problem is not simply that something is wrong psychiatrically? What if the problem is deeper? What if a metabolic pathway, a transport system, or an energy pathway inside the brain is not working correctly? That is when doctors must think about inborn errors of metabolism, or IEMs. These disorders are easy to miss, because sometimes they look psychiatric. They can look like psychosis. They can look like autism. They can look like epilepsy. They can look like developmental delay. They can look like a behavioral problem. So the next step may be to search for metabolic clues inside the body. Doctors may order a genetic and metabolic workup. Is the ammonia high? Is the blood sugar low? Is lactate elevated? Are the electrolytes abnormal? Are there problems in the blood gas, ketones, clotting studies, or liver function? Are there abnormalities in urine organic acids, plasma amino acids, or acylcarnitines? Are there clues in homocysteine, methylmalonic acid, vitamin B12, or folate? Does the MRI show changes in the white matter, basal ganglia, brainstem, cerebellum, or cortex? And if those tests still do not explain the child's symptoms, doctors may look one step closer to the brain. They may examine the cerebrospinal fluid, or CSF. CSF is like a small sample of fluid from the central nervous system, a kind of central fluid sample near the brain. Some problems are hard to see in the blood, but they may leave clues in the CSF. For example, CSF glucose, CSF lactate, CSF amino acids, 5-MTHF, pterins, and certain metabolites related to dopamine and serotonin. CSF can help doctors study the chemical environment around the brain, but CSF testing is invasive. It is not something doctors do casually. There must be a clear medical reason. The sample must also be collected, frozen, and stored properly. If the sample is handled incorrectly, the result may be affected. There is another test that often appears alongside MRI. It is called Brain-1, HMRS, or Proton Magnetic Resonance Spectroscopy. A standard MRI mostly looks at brain structure. Is there a white matter lesion? Is there injury in the basal ganglia? Is there brain atrophy? Are there abnormalities in the brainstem or cerebellum? But MRS asks a different question. Inside this region of the brain, are certain metabolic peaks normal or abnormal? You can imagine MRS this way. A doctor looks at a map of the brain and circles one small room. Then the doctor listens to the chemical sounds inside that room. Different metabolites make different sounds. On the report, those sounds appear as peaks, and sometimes those peaks are footprints left behind by an inborn error of metabolism. MRS does not require a brain biopsy. It does not require a spinal tap. It is a non-invasive test performed during an MRI scan. Common metabolites include NAA, choline, creatine, myoinositol, GLX, lactate. Under certain conditions, MRS may also detect GABA, glutathione, glycine, and lipids. It sounds like a chemical inventory of the brain, but one thing is critical. MRS is not a universal screening test. A normal MRS does not completely rule out an inborn error of metabolism. Many key molecules are present at very low concentrations. Their peaks may overlap. A child's age matters. The scan settings matter. The sampling location matters. The post-processing method matters. All of these can affect the result. So one peak cannot name a child's disease by itself. MRS is most useful when it is placed inside a larger evidence table. The clinical picture, the MRI pattern, the MRS peaks, blood and urine metabolic testing, CSF results, genetic testing, enzyme studies or functional evidence, and the response to treatment. When these clues are brought together, doctors get closer to the truth. Chapter 1. What is MRS actually looking for? 1. HMRS stands for Proton Magnetic Resonance Spectroscopy. It is not reading what a child is thinking. It is measuring metabolite signals inside a specific region of the brain. For example, NAA. NAA is often used as a marker of neuronal and axonal integrity. Creatine. Creatine is like an energy buffer battery inside brain cells. Choline. Choline is related to cell membrane turnover, myelin, and inflammation. Lactate. Lactate may suggest energy metabolic stress in that brain region. Myoinositol. Myoinositol is related to glial cells and osmotic balance. GLX. GLX reflects signals related to glutamate and glutamine. But MRS cannot see everything. It is better at detecting molecules that are present in higher concentrations and have clearer peaks. Many key molecules in inborn errors of metabolism are too small, too weak, or too easily hidden under other signals. So MRS is useful, but it is not the final verdict. Chapter 2. What clues can MRS provide in certain metabolic diseases? The first example is Canavan disease. This is a leukodystrophy. It is usually associated with variants in the ASPA gene. The enzyme related to this gene helps process a substance called NAA. When that enzyme does not work, NAA can build up. MRI may show white matter abnormalities. MRS may show a markedly elevated NAA peak. That clue can be very directional. It tells doctors this child's white matter problem may be related to the NAA metabolic pathway, but MRS alone cannot confirm the diagnosis. Doctors still need to check urine NAA or perform ASPA genetic testing. The second example is cerebral creatine deficiency syndromes. Creatine is like an energy buffer battery inside brain cells. When this system is disrupted, a child may develop speech and language delay, intellectual disability, seizures, autism-like features, movement abnormalities, or behavioral problems. In these IEMs, MRS can show an important clue. The creatine peak in the brain may be markedly reduced or even absent. That signal can be striking. But MRS cannot tell doctors which specific type is present. Further testing is still needed. Doctors may examine creatine-related metabolites in blood and urine. They may also check guanidinoacetate, enzyme function, or genetic testing. The third example is glycine encephalopathy. It is also called non-ketotic hyperglycinemia. The problem lies in the glycine cleavage system. Too much glycine accumulates in the brain that can disturb the balance between excitation and inhibition. Children may develop severe seizures, low muscle tone, apnea, and developmental impairment. In some cases, MRS may show an elevated glycine signal, but the glycine peak can easily overlap with other signals, so MRS cannot diagnose it alone. Doctors must also examine plasma glycine, CSF glycine, the CSF to plasma glycine ratio, and they must combine this with urine organic acids, acetocarnitines, and genetic testing. The fourth example is mitochondrial disease and the Lee spectrum. Mitochondria are the power plants of the cell. When the power system fails, the brain may not get enough energy. Pyruvate may not move smoothly into normal energy metabolism. More of it may be converted into lactate. At that point, MRS may show a lactate peak. What is a lactate peak like? It is like smoke rising from a power plant. It suggests that this brain region may be in an energy crisis. But seeing smoke does not immediately tell you which machine is broken. A lactate peak is not specific to mitochondrial disease. Hypoxia, the period after seizures, infection, tumors, and necrotic tissue can also produce lactate, so doctors have to keep investigating. This becomes especially important when a child has lesions in the basal ganglia, brainstem, or cerebellum, or when a child suddenly regresses after infection, fasting, surgery, or anesthesia. In those cases, energy metabolism must be taken seriously. Chapter 3. Why is standard MRS not enough? Because many key molecules in inborn errors of metabolism are very small, their concentration is low, their signal is weak, and their peaks can be crowded against other peaks. Take serine, for example. Serine is very important for brain development. It participates in phospholipid metabolism, sphingolipid metabolism, 1-carbon metabolism, and folate pathways. It also participates in the production of D-serine. When serine synthesis is impaired, a child may develop microcephaly, refractory seizures, developmental delay, white matter abnormalities, peripheral neuropathy, ataxia, or behavioral problems. But here is the problem. Serine concentration in the brain is low. The signal is weak. The peak overlaps easily. On standard MRS, serine is like a small child standing in a crowded room. Its voice is quiet, and several people nearby are speaking much louder. So at this time, brain 1H MRS is not a standard diagnostic method for serine deficiency disorders. The more reliable path is still clinical evaluation. Doctors check plasma and CSF serine. They compare the results with age-specific reference ranges. They perform genetic testing for PHGDH, PS81, and PSPH, and when needed, they combine this with functional evidence and the response to L-serine treatment. But research has not stopped. There are already research efforts using 7T ultra-high-field MRS. Scientists are trying to pull out the weak serine signal from the overlapping chemical noise. That tells us something important about the future. The goal is to hear the small signals we cannot clearly hear today, one by one. Chapter 4. J-edited MRS, pulling small molecule signals out of a crowd of noise. What is standard MRS like? It is like recording every sound in the emergency room at the same time. Doctors are talking, nurses are talking, machines are beeping, family members are speaking. If the molecule you want to hear has a very weak voice, it can easily be drowned out. J-edited MRS uses a different way of listening. It uses special pulses to change the signal of the target molecule. Then it subtracts one spectrum from another. This cancels out much of the background signal. What remains is the small signal the doctor or researcher is trying to see. One of the most common techniques is called megapress. It is often used to study GABA, glutathione, lactate, glycine, and 2-hydroxyglutarate. Some studies also try to examine low-concentration amino acids. GABA is especially important here. It is the brain's major inhibitory neurotransmitter. You can think of it as the brain's braking system. If the brakes are unstable, neural networks may become overexcited. At 3T field strength, megapress is one common method for measuring GABA+ or GABA+. In recent years, expert guidelines have also provided specific recommendations for GABA megapress: how to acquire the data, how to perform quality control, how to process the data, how to report the results. That shows how important the method is. But it also shows something else. This is not a test that can be done casually or interpreted casually. For inborn errors of metabolism, J-edited MRS has practical value. It may help researchers see small molecules that standard MRS cannot separate well. For example, neurotransmitters, redox-related molecules, and low-concentration metabolites. This may be useful for studying GABA metabolism disorders, glutathione-related oxidative stress, glycine encephalopathy, and lactate-related energy abnormalities. But we have to remember, right now, J-edited MRS is still mostly a research tool, or a tool used in advanced medical centers. It is not yet a routine clinical screening test for inborn errors of metabolism. Chapter 5. 7T MRSI and 2D MRS. Drawing a sharper metabolic map of the brain. First, let's talk about 7T ultra-high field MRI. Most hospital MRI scanners are 1.5 Tesla or 3 Tesla. But 7 Tesla has a much stronger magnetic field. It is like using a more sensitive recording device. It may hear finer chemical sounds, and it may separate metabolites that are crowded together. For example, glutamate, GABA, glutathione, myo-inositol, and NAA. But 7T also has real-world limits. There are not many scanners. Children may have trouble cooperating. The scan parameters are complex. The high magnetic field itself creates technical challenges. Normal reference ranges are still limited, and different medical centers may not be fully standardized. So for now, 7T is more of a future direction. It is not yet an everyday clinical tool for diagnosing inborn errors of metabolism. The second technology is called MRSI or CSI. MRSI stands for Magnetic Resonance Spectroscopic Imaging. CSI stands for Chemical Shift Imaging. Standard single voxel MRS looks at only one small area of the brain. It is like listening to one room, but MRSI can examine many brain regions at once. It can create a larger metabolic map. That matters for IEMs, because many inborn errors of metabolism do not injure only one spot. Mitochondrial disease may affect the basal ganglia, brainstem, cerebellum, and cortex. Leukodystrophies may spread from deep white matter toward subcortical white matter. Hyperammonemic encephalopathy and organic acidemias may also involve multiple brain regions. So sometimes looking at one small point is not enough. Doctors and researchers need to see the wider metabolic pattern. In recent years, whole brain 3D1 HMRSI has been used in human brain metabolic connectivity research. Researchers may study metabolites such as TNAA, TCR, GLLX, myoinositol, and choline. They look for metabolic similarities across different brain regions. The third direction is 2D MRS. Standard MRS is a one-dimensional spectrum. Many metabolites are crowded along one line. It is like many people standing in the same row and speaking at once. 2D MRS spreads the signals out. It can make some crowded chemical voices easier to separate. For example, glutamate, glutamine, GABA, myoinositol, glutathione, lactate, and some amino acids. Research on 2DJ resolved MRS has shown that it can improve the separation of human brain metabolites. It can also help assess how reproducible different metabolite measurements are. In simple terms, the future of brain metabolic imaging may not be just one peak on one spectrum. It may be a much more detailed metabolic map of the brain. Chapter 6. MRS looks at brain regions. CSF looks at the central nervous system environment. If MRS is like listening to metabolic sounds inside one room of the brain, then cerebrospinal fluid, or CSF, is like taking a small fluid sample near the brain. CSF flows around the brain and spinal cord. It helps cushion them. It participates in metabolic exchange, and it helps maintain the environment of the central nervous system. A CSF test is not a brain tissue biopsy. It cannot represent every region of the brain, but it is closer than blood to the environment around the brain and spinal cord. Some clues may be hard to see in blood or urine, but they may appear in the CSF. In clinical neurometabolic evaluation, CSF testing may include CSF glucose, CSF lactate, CSF amino acids, CSF5-MTHF, CSF pterans, and neurotransmitter metabolites, for example, HVA, 5-HIAA, MHPG, 3-OMD, LDOPA, and 5-HTP. These tests can answer questions that MRS cannot. Can glucose enter the brain? Can folate reach the central nervous system? Is there a problem in the BH4 cofactor pathway? Are there breaks in dopamine, serotonin, or norepinephrine A pathways? Are glycine, serine, asparagine, or glutamine abnormal? Is there a lactate clue pointing toward central energy metabolism? Testing CSF monoamine metabolites, 5-MTHF, and pterines is already used in the evaluation of some neurotransmitter disorders and related neurometabolic diseases. But CSF testing also has strict requirements. Sample handling matters. Age matters. Medications matter. The collection process matters. All of these can strongly affect how the results are interpreted. So MRS and CSF do not replace each other. They are two different kinds of evidence. MRS tells doctors which brain region may have abnormal metabolism. CSF tells doctors which metabolites may be abnormal in the central fluid environment. Blood and urine metabolic testing tells doctors whether the body's metabolic pathways are leaving traces. Genetic testing tells doctors which biochemical blueprint may contain an error. When these clues are brought together, some cases that look like psychosis, autism, epilepsy, or developmental delay can be pulled back toward a specific biochemical pathway. Chapter 7. Why might psychiatric symptoms be connected to brain region metabolism? Psychiatric symptoms may look like problems of thought, emotion, and behavior, but the brain is not a psychological machine floating in the air. It is a high-energy city. The frontal lobes help with planning, judgment, and impulse control. The temporal lobes participate in language, memory, and auditory processing. The limbic system is involved in fear, emotion, and reward. The basal ganglia help regulate movement, motivation, and the initiation of action. The cerebellum does more than balance. It also participates in cognition and emotion. The brainstem and autonomic networks influence sleep, breathing, heart rate, and alertness. For these brain regions to work properly, several basic conditions must be met. There must be enough energy. Mitochondria must be able to generate power. Neurotransmitters must be made, and they must also be cleared. Ammonia, lactate, organic acids, and abnormal amino acids must not poison neurons. Cofactors such as 5-MTHF, BH4, vitamin B6, and vitamin B12 must be available, and the excitatory-inhibitory balance between glutamate and GABA must not stay out of control. If one pathway becomes blocked, the brain may not go into coma immediately. The first signs may be more subtle: poor attention, emotional instability, disrupted sleep, hallucinations, anxiety, migraine, autism-like features, aggressive behavior, loss of learning skills, seizures, ADHD-like symptoms, or abnormal movements. So some psychiatric and behavioral symptoms may be related to brain energy metabolism, mitochondrial function, redox status, and the glutamate GABA system. This does not mean that all psychiatric illness is metabolic disease. It means something more careful. When psychiatric symptoms appear alongside neurologic and metabolic red flags, doctors need to look deeper. Chapter 8. What is the core question in metabolic psychosis? Does the brain lack fuel? Is a pathway blocked? Are toxic substances building up? Metabolic psychosis refers to psychiatric symptoms related to inborn errors of metabolism. It can also mean psychotic features caused by inherited metabolic disease. You can think of it as a chemical traffic accident inside the brain. Some roads are fuel roads. Glucose cannot enter the brain. Fat cannot be burned properly. Mitochondria cannot generate enough power. Some roads are detoxification roads. Ammonia cannot be cleared. Organic acids build up. Lactate rises. Some roads are signaling roads. Dopamine, serotonin, GABA, or glutamate cannot be made or cleared properly. Some roads are cofactor roads. When 5-MTHF, BH4, PLP, or vitamin B12 is not sufficient, a chain of enzyme reactions may slow down. And some roads are membrane and myelin roads. Abnormal metabolism of NAA, choline, myo-inositol, or lipids may affect white matter and neural networks. These problems do not always appear as coma. They do not always look like a classic metabolic crisis. They may also appear as psychiatric or behavioral changes. For example, abnormal dopamine pathways may affect movement, motivation, reward, and psychotic symptoms. Abnormal serotonin pathways may affect sleep, mood, the gut, pain, and behavioral regulation. Low GABA, or excessive glutamate excitation, may affect seizures, anxiety, sensory overload, and network stability. Unstable mitochondrial energy supply may cause fatigue, cognitive fluctuation, brain fog, episodic regression, exercise intolerance, and emotional collapse. High ammonia or organic acid accumulation can cause delirium, hallucinations, altered consciousness, and aggressive behavior. In clinical practice, this can easily be mistaken at first for acute psychosis. This is where the future promise of MRS plus CSF becomes especially important. Together, they may help doctors map the brain metabolic state behind psychiatric symptoms. The question should not only be, does this person have psychiatric symptoms? Doctors also need to ask, Are glutamate and GABA abnormal in the prefrontal cortex? Is energy metabolism in the basal ganglia adequate? Do NAA or choline changes in the white matter suggest neuronal injury or myelin injury? Do CSFHVA and 5-HIAA suggest abnormalities in dopamine or serotonin pathways? Does CSF5-MTHF suggest cerebral folate deficiency? Do CSF lactate and MRS lactate peak and 31P energy markers point together toward mitochondrial energy dysfunction? These are the questions future research on metabolic psychosis must explore. The goal is not to diagnose every psychiatric symptom as metabolic disease. The goal is to find, in people with red flags, the brain metabolic crises that may otherwise be missed. Chapter 9. Psychosis, depression, autism, epilepsy, and developmental delay may one day be classified in a more precise way. But we have to be careful here. Not every case of schizophrenia is a metabolic disease. Not every case of depression is mitochondrial disease. And not every child with autism can be helped by metabolic treatment. The real point is this. The same symptom can come from different causes. Take psychotic symptoms, for example. In one person, they may be related to immune dysregulation. In another, they may be linked to head trauma, a brain infection, or a tumor. In someone else, they may come from medications, alcohol, or toxins. And in some people, they may come from an inborn error of metabolism, or IEM. For example, a urea cycle disorder. Glycine encephalopathy, CBLC disease, MTHFR deficiency, Niemann-Pick disease, type C, or NPC, mitochondrial disease, or a neurotransmitter disorder. The same is true for depression. In one person, depression may be related to inflammation, hormones, or sleep. In another person, it may involve energy metabolism. And in a smaller group of patients, there may be clues involving folate, vitamin B12, 1-carbon metabolism, mitochondrial function, or redox balance. The same is true for autism-like features. If autism-like symptoms come together with seizures, developmental regression, abnormal movements, low muscle tone, oculogeric crises, white matter abnormalities, or multi-system involvement, doctors cannot stop at the word autism. They also need to include inborn errors of metabolism in the differential diagnosis. MRS has already been used in psychiatric research. Researchers have studied brain metabolites such as glutamate, GABA, NAA, lactate, and glutathione. In schizophrenia research, there has also been long-standing interest in the glutamate system, the GABA system, and abnormalities in energy metabolism. But this point is important. Most of this research is aimed at understanding mechanisms and identifying subtypes. It does not mean that MRS can be used clinically to diagnose psychiatric illness directly. Chapter 10. The Future Metabolic Map of the Brain. New technologies are now helping researchers look deeper. The first is DMI, or deuterium metabolic imaging. A simple way to understand it is this. DMI looks at how the brain uses sugar. Researchers can attach a safe label to glucose. Then they can observe what happens after that glucose enters the brain. Does it become lactate? Does it become glutamate? Does it become glutamine? Or does it enter another metabolic route? For inherited metabolic diseases and metabolic neuropsychiatric disorders, DMI asks very direct questions. Is the brain getting fuel? After the fuel enters, can it be used to generate energy properly? Which brain regions are more likely to lose power? In the future, conditions such as GLUT1 deficiency, mitochondrial disease, pyruvate metabolism disorders, seizure foci, and regions of developmental regression may all need this kind of metabolic map. But for now, DMI is still mainly a medical research tool. The second technology is carbon-13, or C13, and hyperpolarized C13. You can think of it as placing a carbon label on metabolic fuel. Researchers can then watch where glucose or pyruvate goes after it enters the brain. Does it move toward lactate, toward glutamate, toward glutamine, toward GABA, or does it enter the mitochondrial energy cycle? The more advanced version is hyperpolarized 13C. It can greatly amplify the 13C signal. That helps researchers see the fate of pyruvate much more clearly. Pyruvate is important. It is like a major intersection in metabolism. If pyruvate moves toward lactate, that may suggest increased glycolysis or greater reductive stress. If it moves toward acetyl-CoA, it enters the mitochondria and the energy cycle. If it moves toward alanine, it is connected to amino acid metabolism. If it becomes bicarbonate, that is related to PDH, or pyruvate dehydrogenase, and oxidative metabolism. Many metabolic encephalopathies and mitochondrial diseases get stuck near this intersection. In the future, if this technology becomes safer, faster, and easier to use, it may help researchers study brain energy abnormalities in psychosis risk states. It may help study lactate and oxidative metabolism changes in depression or bipolar disorder. It may help ask whether an epileptic focus has abnormal glycolysis. It may help show which brain regions do not receive enough energy in mitochondrial disease. And it may help researchers see whether treatment changes the direction of pyruvate flow. The third technology is 31-PMRS, or phosphorus 31-MRS. If 1-HMRS looks at metabolic fingerprints such as NAA, lactate, choline, and creatine, then 31-PMRS is more like looking at the cell's energy ledger. It can examine ATP, phosphocreatine, inorganic phosphate, intracellular pH, and other phosphorus signals related to energy metabolism. For inherited metabolic encephalopathies, 31-PMRS asks questions like these. Is there enough ATP? Are phosphocreatine reserves adequate? Is the cell's acid-base state abnormal? Can mitochondrial power production keep up with the demands of that brain region? This may help researchers explore important clinical questions. Why do some patients have long-term fatigue, brain fog, and worsening after exercise? Why do some children suddenly regress after infection? Why might certain subtypes of psychosis or depression involve abnormal brain energy metabolism? And after treatment, does the brain's energy state improve? For that reason, 31-PMRS may be especially useful in future research on mitochondrial disease, energy metabolism disorders, neurodegeneration, and some difficult neuropsychiatric symptoms. The fourth technology is CEST and Glucine. CEST stands for Chemical Exchange Saturation Transfer. It is not a traditional MRS spectrum. Instead, it uses proton exchange between molecules and water to create indirect metabolic-weighted images. GluCEST mainly looks at glutamate-related signals. Glutamate is the brain's main excitatory neurotransmitter. It is also the precursor for GABA, and it is connected to energy cycling and the metabolic partnership between neurons and glial cells. In the future, GluCEST may help researchers study glutamate signals in epilepsy, psychosis, depression, and neurodevelopmental disorders. If glutamate activity is too strong, neural networks may become overexcited. If GABA inhibition is too weak, the risk of seizures and sensory overload may increase. If the glutamate-glutamine cycle is abnormal, that may suggest a problem in the cooperation between neurons and glial cells. In hyperammonemia, glutamine can build up. That can contribute to brain swelling and neurotoxicity. If CEST technology becomes more mature, it may help doctors see which brain regions have abnormal glutamate signals, whether there is metabolic excitation around a seizure focus, whether certain subtypes of psychosis or depression involve specific brain region abnormalities, whether children with autism-like regression have a distinct neurotransmitter metabolic pattern, and whether glutamate signals come down after treatment. But right now, CEST is still affected by many factors: pH, magnetic field homogeneity, water content, and the tissue environment. So it is promising, but for now it remains mainly a research tool. The fifth direction is multinuclear MR and metal-sensitive imaging. The brain contains more than sugar and lactate. It also depends on ion balance, and it can carry traces of metal deposition. Standard 1, HMRS, cannot directly measure copper, iron, manganese, or zinc the way a CSF metal test might. It does not simply give you a copper peak or an iron peak. More often, it shows metabolic traces left behind after metal balance or ion homeostasis has affected brain cells. For example, a decrease in NAA, a change in choline, the appearance of a lactate peak, or an abnormal energy ledger on 31 PMRS. The technologies that come closer to an ion window are mostly multinuclear MR research methods. For example, 23NA MRI can study sodium concentration in brain tissue. It is like asking whether the brain cell's saltwater pump, which maintains the sodium gradient, is losing balance. 39K MRI is also being explored for potassium in the brain, but it is still at an earlier stage. 31 PMRS can also provide indirect information about free magnesium ions and energy metabolism. For metal deposition, the more mature tools in clinical care and research are often not MRS. They are MRI techniques sensitive to magnetic susceptibility and relaxation. For example, iron deposition can be studied with SWT2 star, R2 star, and QSM. This is important in NBIA, neurodegeneration with brain iron accumulation, and in some other neurodegenerative disorders. Manganese overload often affects the basal ganglia. A classic clue can be T1 hyperintensity in the globus pallidus. In some manganese transport disorders, this is a very important imaging clue. Wilson disease is a disorder of copper metabolism. Brain MRI and QSM can help observe injury and magnetic susceptibility changes in regions such as the basal ganglia, thalamus, and brainstem. But diagnosis cannot replace ceruloplasmin testing, urine copper testing, an eye examination, and ATP7B genetic testing. So CSF metal testing is like taking a small sample of central nervous system fluid and asking which metal elements are present. Brain MR and MRS are more like examining the scene inside the brain. Which brain region has changed signal? Where might iron or manganese be deposited? Which energy ledger looks abnormal? Which metabolic changes may be injuring brain regions? These tools can complement each other, but many of them are still in the research stage. Chapter 11. What matters most in the future is connecting the evidence into a pathway. The goal of these future tests is not to make every patient undergo more testing. The goal is to connect the clues into a meaningful route. In the past, a patient might have been described as having psychosis, autism, epilepsy, developmental delay, cerebral palsy, or severe treatment-resistant behavior problems. But in the future, doctors may keep asking, Are glutamate and GABA abnormal in the prefrontal cortex? Is there lactate or abnormal energy metabolism in the basal ganglia? Is CSF5-MTHF low? Do HVA and 5-HIAA suggest a problem in monoamine pathways? Does 31-PMRS suggest abnormal ATP or phosphocreatine? Does DMI show abnormal glucose utilization? Does 13C suggest that pyruvate cannot enter oxidative metabolism smoothly? Does CEST show an abnormal glutamate map? Can genetic testing identify a matching metabolic disease clue? And after treatment, do these abnormalities improve? This could move some patients from being named by symptoms to being classified by mechanism. Here are a few examples. If psychotic symptoms come with high ammonia or abnormal glutamine, doctors should consider a urea cycle disorder or hyperammonemic encephalopathy. If depression or psychiatric withdrawal comes with high homocysteine, low methionine, or a 5-MTHF problem, doctors must first rule out common acquired causes. Then they may consider one-carbon metabolism, vitamin B12, MTHFR, CBLC, and related pathways. If autism-like features come with oculogeric crises and abnormal CSF monoamines, doctors should consider AADC deficiency, tyrosine hydroxylase deficiency, BAH4 metabolism disorders, and related conditions. If epilepsy comes with a lactate peak and basal ganglia abnormalities on MRI, doctors should consider mitochondrial disease or an energy metabolism disorder. They still need to continue with genetic testing, biochemical testing, and evaluation for treatable causes. If developmental delay comes with an absent creatine peak, doctors should consider cerebral creatine deficiency syndromes. The next step is to check creatine-related metabolites in urine and blood. Doctors may also examine guanidinoacetate, the creatinine ratio, and the related genes. If behavioral regression comes with white matter abnormalities and abnormal findings on MRS or CSF testing, doctors should consider leukodystrophy, lysosomal disease, organic acidemia, or another inherited metabolic disease. This is the most promising part of metabolic psychosis research. The point is not to call every psychiatric illness a metabolic disease. The point is to look underneath complex psychiatric and neurologic labels and identify the IEMs that can be tested, explained, and in some cases, treated. Chapter 12. Several clinical cautions matter right now. First, MRS and CSF cannot diagnose most psychiatric disorders by themselves. They provide metabolic evidence. They are not replacements for psychiatric diagnosis. Their real value appears when a patient has red flags. They can help doctors re-examine symptoms that look like psychosis, autism, or epilepsy through the lens of brain regions, metabolites, transport pathways, and genetic causes. Second, diagnoses such as psychosis, depression, autism, epilepsy, and developmental delay describe clinical presentations and syndromes. They do not always identify the true cause. Third, CSF testing is invasive. Whether a lumbar puncture is needed must be decided by neurology and metabolic genetics specialists based on the patient's condition. Fourth, DMI, 13C, 31P, and CEST are still mostly research tools or advanced center technologies. They are promising, but at this time they cannot be used for routine screening of psychosis, autism, or depression. Fifth, any metabolic abnormality must be interpreted in the context of the patient. One peak, one CSF marker, one genetic variant. None of these can name a disease when separated from the clinical picture. Blood and urine metabolic testing, enzyme and functional studies, genetic testing, and the patient's symptoms belong on the same evidence table. Conclusion. The future of psychiatric research may include a new kind of map, a metabolic map of the brain. For many families, the most painful part is not hearing a complicated diagnosis. It is watching a child get worse while receiving only vague explanations again and again. It looks like epilepsy. It looks like autism. It looks like psychosis. It looks like anxiety. It looks like cerebral palsy. It looks like a teenage phase. The future value of MRS and CSF is to keep following those words. Looks like. These tools push doctors to ask deeper questions. Which region of the brain is running out of fuel? Which neurotransmitter production line has broken down? Which metabolite is building up? Which cofactor cannot reach the brain? Which brain region cannot keep up with ATP demand? Which network is being pulled by an imbalance between glutamate and GABA? And after treatment, does the metabolic pattern begin to turn back? This will not solve every psychiatric or neurologic disease, but it may change the future for some patients, especially children and adults with red flags. Developmental regression, treatment-resistant seizures, abnormal movements, oculogeric crises, worsening after infection, fasting, exercise, or certain medications. Multi-system involvement, white matter or basal ganglia abnormalities, family history of unexplained death or similar symptoms, psychiatric symptoms surrounded by clear neurologic or metabolic clues. These patients should not be covered over by one psychiatric label. Future genetic and metabolic testing may help place them on a clearer map. Blood tells us about the body's metabolic traffic. Urine tells us which abnormal waste products the body is trying to eliminate. CSF tells us about the chemical environment of the central nervous system. MRS tells us about metabolic peaks inside brain regions. DMI, 13C, 31P, and CEST are still more like research telescopes. They help researchers observe how fuel flows, how energy is produced, how glutamate is distributed, and whether mitochondria can keep up. This map is not finished yet, but the direction is becoming clear. Behind psychiatric symptoms and neurologic symptoms, we must keep asking about the brain's chemical reality. For inherited metabolic encephalopathies and metabolic psychosis, this may become one of the most important research gateways of the next several decades. Disclaimer: this video is for medical education only. It cannot replace diagnosis or treatment by a physician. Whether a patient needs brain MRS, CSF testing, blood and urine metabolic testing, genetic testing, or enzyme and functional studies should be decided by pediatric neurologists, metabolic genetics specialists, psychiatrists, and neuroradiology specialists, based on the specific clinical situation.
If you are interested in inborn errors of metabolism, or IEMs, and in the connection between neurologic or psychiatric symptoms and inherited metabolic encephalopathies, you can find the full written version in the Shunyi series.